Welcome back to yet another video of Edu Learning Platform. Today we will learn about gas chromatography and HPLC column chromatography in detail. It is very essential for FSO exam and fssai conducted exam by government, central government. It is very useful for many science oriented exam. Nowadays questions can be asked from anywhere, so just you study all this chromatography technique in detail. So we directly move to chromatography. That is, chromatography can be done in a tube. That means it can be referred as column chromatography, and there is a little bit different from paper chromatography and thin layer chromatography that we will discuss later. Then just you go through the chromatography. Then gas chromatography can be done the column. The tube can be packed column or capillary column. Packed column is made up of glass or stainless steel, whereas capillary column is made up of material fused, that is material fused quartz material. So we have to study more detail about gas chromatography. Gas chromatography, the column is based in a chamber so that uniform temperature can be maintained. This stationary phase is made up of silicon, grease or wax and kept in inner space of the column. Whereas the stationary phase, which can be withstand high temperature, and mobile phase is used like helium or unreactive gas such as nitrogen. The mobile phase gas is kept in the cylinder, which is connected with column by a molecular sieve. Molecular sieves separate unwanted hydrocarbon, water vapor and oxygen that may interface with the sample during analysis. At the end of the column, there is a detector that detects the sample. The sample is mixed with volatile solvent like heptane, acetone, methanol, etc. So the name of the detector is very important. We use the detector, that is flame ionization detector. So just above the spectrum to inject the sample can be placed. The temperature of the injection region is kept 20 to 50 degrees Celsius higher than the column. This allows rapid visualization of the sample. During the analysis, the temperature is kept up to 150 to 300 degrees Celsius. So just you note the correct setup of gas chromatography. Detector is used, and from detector we get the sample is detected the computer and give the corresponding peak. Then we can identify which one it is, and separation occurs interaction of molecular between the stationary phase and mobile phase. Less volatile molecule interact more with the stationary phase and go slowly, and mobile phase move fast down the column. Stationary phase may go slowly. Less volatile molecule it's very move slowly. Gas chromatography is a common technique of chromatography used in the analytical chemistry for separating and analyzing compound that can be vaporized without decomposition. In simple sense, we have to understand gas chromatography is used to separate the volatile compound. Next, we move to Absorption chromatography. Adsorption chromatography is a one type of Coulomb chromatography, and for that we have to study about Coulomb chromatography first. Coulomb chromatography, the some new words are there in Coulomb chromatography. First one is mobile phase can be referred as eluent, and solvent leaving column can be named as eluate, and sample leaving column can be known as eluite. These three terms are included in the Coulomb chromatography. So adsorption Coulomb chromatography is a separation technique. The Coulomb chromatography is a separation technique in which the stationary bed is within a tube. So stationary bed is arranged in a tube. When it is the stationary phase is silica or alumina, it can be referred as adsorption chromatography. The sample mixture is a solvent is loaded on the Coulomb. The individual compound get differentially absorbed on the absorbent. Elution is carried out by the buffer system, which is mobile phase. Coulomb chromatography, the mobile phase we call it as eluent. Sample leaving column is eluite, and mobile phase leaving column is eluate. And the setup used in the Coulomb chromatography, is like this. We can arrange the mixture in a tube, and the down it can be used, cotton can be used, and other form of Coulomb chromatography. Whether the stationary phase is silica or alumina, it call it as adsorption chromatography. The stationary phase is ion exchange resin, we call it as ion exchange chromatography. When it is ligand, it known as affinity chromatography, and when it is porous beads or gel, the gel permeation chromatography. Next we move to HPLC, high performance liquid chromatography. Liquid chromatography is a separation technique in which the mobile phase is a liquid. Liquid chromatography can be carried out either in a column or a plane. Present day liquid chromatography that generally utilizes very small packing particle and relatively high pressure. So we call it as HPLC. HPLC, the sample is forced by a liquid or a high pressure through column that is packed with a stationary phase composed of irregularly or spherically shaped particle. The interaction between mobile and the stationary phase leads to the separation of the mixture. So let's see what happened in the HPLC. HPLC means high performance liquid chromatography. So as far as the HPLC is concerned, HPLC is also known as high performance liquid chromatography. Here the Coulomb is made up of stainless material, and stationary phase can be absorbent material. So here look, column internal diameter is 4.5 millimeter and length is about 5 to 25 centimeter, and it is made up of stainless steel. Stationary phase is an adsorbent material. Very small particles like chemically modified silica, divinely benzene, etc. can be taken as stationary phase. So what about the mobile phase? Mobile phase is a solvent reservoir, can be polar or non-polar solvent, depends on the type of sample. HPLC can be referred as high performance liquid. High pressure pump is placed above the column, and it may give a certain amount of pressure, and above the column there is a sample injector, and after separating the mixture it will connect it to a detector, and the detector will give the corresponding P of the material. Normal phase HPLC is there, where the stationary phase is polar and mobile phase is non-polar. And here also the specialty is that stationary phase is polar and mobile phase is non-polar. This is known as normal phase HPLC. And next one is that polar molecule are returned in the column. Non-polar molecule moves down the column with mobile phase. The specialty in normal phase: polar molecule return in the column, non-polar molecule moves down the phase. The non-polar molecule in reverse phase HPLC, non-polar molecule are returned in the column, whereas polar molecule moves down the column with the molecular phase. And there are different types of HPLC we are discussing, that is normal phase HPLC, reverse phase HPLC, and size exclusion HPLC. Here also we note that there are some porous beads are included in it. Another type of HPLC is ion exchange HPLC. Here it is based on the charge. When the sample is negative, the molecule of interest with taken place is plus charge. So different types of HPLCs are there: normal, reverse, and ion exchange. So as far as one important thing is that detector, UV detector, IR detector, refractive index detector, mass spectrometer, electrochemical detector, these all are used in HPLC. So HPLC is considered as far as HPLC is considered, is called high performance liquid chromatography. HPLC is usually modified column chromatography. In this column, it's kept in the absorbent is placed on a material is like silica, and mobile phase is placed in the above the stationary phase. Because of the gravity, a high pressure pump is attached with the column. Stationary phase is silica or aluminum, and the column is filled with cellulose absorbent material, and the separation take place, and when the separation take place, it can be detected to a detector: UV detector, IR detector, refractive index detector, mass spectrometer, electrochemical detector can be used in this process. So the name of the detector you have to study. Okay, thank you for watching this video. Can like, share, and subscribe. The question is asked in objective ways that you have to study the objectives. You have to study all the theories and all. I'm very glad to explain in the simple English only. It's very nice to explain, and we have to understand the explanation of English. So keep